All right, and the last bit of this lecture are the gram-positive bacteria that are bacilli or rod-shaped, and they do not make endospores. So anything that's not a bacillus or a clostridium, that's gram-positive and rod. And there are 4 main genus or genera of bacteria that fall into this category: the Listeria, the Mycoplasma, the Corynebacterium, and the Mycobacterium. So we're going to start first with the Listeria. Of all the Listeria that are out there, it's really only the monocytogenes that causes issues with humans. So generally when you hear about a Listeria outbreak or a Listeria infection, it is specifically Listeria monocytogenes. Sometimes they just shorten it and call it L-mono, just like we call it, instead of constantly saying Clostridium difficile, we call it C. diff. A lot of times we just call this L-mono. When we gram stain it, again, it is gram-positive, and it's bacilli, but they're very, very short rods, almost round. So we call it a coxobacillus shape. Because they're not perfectly round, we can't call them a coccus. So we kind of smush the two words together. It's not quite coccus, it's not really bacillus, it's a coxobacillus. Where we pick them up when we ingest contaminated food or any kind of beverage that it might be found in. This particular bacteria usually resides in soil as well as the digestive system of animals usually. So we're pretty much eating contaminated food from either fecal matter or from soil that had fecal matter in it. Its big claim to fame that makes it so virulent or able to cause disease is that it is an intracellular parasite. So intracellular means this thing lives inside of our cells, and it doesn't get broken down, which is awesome. Like, it actually encourages macrophages, the big eating cells part of our immune system. It encourages these cells to get eaten. It would like to get eaten because it's figured out a way not to get broken down and digested by these cells. And so it lives very well protected inside of these cells, almost hiding in our own immune system. And so it generally lives in either macrophages, cells found in the liver, or cells found in the gallbladder. It's a nice tricky way for it to survive. Now, for those that have healthy immune systems, usually doesn't cause us a lot of issues. Usually it may lead to some diarrhea, kind of food poisoning type symptoms if you ate or drank something that had this bacteria. The biggest issue is that this bacteria can cause meningitis in fetuses, so pregnant women have to be very careful with this bacteria, and immunocompromised, so elderly, sick individuals usually suffer the most severe complications. So if you ever hear Listeria outbreaks in the news, which I included an article, usually they're worried about elderly, immunocompromised, and pregnant women. And it's because if you're pregnant and you pick up this bacteria, it is a bacteria that can cross the placental barrier. Most of them cannot, and it can lead to stillbirths, miscarriages, or birth defects. So it can be very deadly. Now, when we diagnose it, we can grow it up. We can also look in a broth for it underneath the microscope because it is flagellated and it likes to tumble around. It does little somersaults underneath the microscope. Treatment, we do have lots of antibiotics that are effective. Again, the sooner you can get treated, the better. I mean, the best is just to not get it in the first place. So avoid certain at-risk foods. And so a lot of these foods are things that even pregnant women are encouraged not to eat while pregnant. So the things that usually have the highest chance of having these bacteria are usually the ones that are targeted in recalls: Brussels sprouts, deli meats and hot dogs, raw milk, which I don't know why you'd want to drink that either, various soft cheeses like brie, and smoked seafood. Although they put this on here because it was 2015 that Bluebell ice cream had a huge outbreak of Listerias. Our next group of bacteria, our next genus that's gram-positive and bacilli that can't make endospores, are the Mycoplasmas. They are the smallest free-living microbes because they can go just hang out in the environment. Kind of its uniqueness is that they are pleomorphic, which means they can change their shape. So they are never perfectly round, so we do not give them that coccus shaped. They're usually bacillus, but then even a varying shaping of bacillus. Part of the reason what makes them able to change shape is that they can't, for whatever reason, make peptidoglycan that's found in the cell wall. So even if we gram stained them, even though they are genetically a gram-positive bacteria, they would actually show up as gram-negative because they don't have that thick peptidoglycan to hold on to the that crystal violet, even though they are genetically gram-positive. To grow them, they are a little picky. They're a little harder to grow. They grow really slow because they do require various growth factors to grow. They usually target the mucous membranes of the respiratory and urinary tracts, leading to pneumonia and urinary tract infections. Probably the most common of the mycoplasmas that causes issues with us is Mycoplasma pneumoniae. It's the only mycoplasma I'm even going to talk about. And it causes what's known as atypical pneumonia. It's more commonly called, when people talk about it, walking pneumonia. And we say it's atypical because we don't have that big immune system response where we'd see a big increase in white blood cells, indicating that there's an infection. It's usually very mild, meaning it's not a pneumonia that you're staying at home thinking you're going to die or even have to be hospitalized. You're walking around because it's so mild. It is spread by nasal secretions, and because kids are dirty, the top grouping of individuals that usually pick this up are kind of children and adolescents. Again, they blow their nose, they don't wash hands, they're touching, they touch everything. Not exactly the most hygienic group of organisms. To diagnose, we usually use an ELISA. That is the fastest way. If we tried to grow it out, it would take forever. They're really slow to grow and they just require various growth factors. Then instead of just, okay, throw it on a plate, blood agar plate for 24 hours in the incubator, it might take 2 to 6 weeks for it to finally grow up enough in a lab to even identify it. So we now just do ELISAs looking specifically for the antigens. Easiest way to not get it, good hygiene, is washing hands, especially after blowing your nose. Our third genus of gram-positive bacilli or rods that do not make endospores is the Corynebacterium. When we gram stain, they are also pleomorphic, and a lot of bacteria are in this genus. There's a lot of species. Most of them don't cause disease, so they're non-pathogenic, and they're just normal skin and oral flora, so we don't care about them because we are focusing on the ones that cause disease. Interesting enough, so they are pleomorphic, so they're not exactly the exact same size and shape all the time, and because they are part of the normal oral flora, it's not uncommon when we do that throat culture that if you see some gram-positive bacilli, but they kind of vary in their shape a little bit, some short rods, some long rods, you probably identified some of your throat culture to a Corynebacterium. That's normal. That one species that does cause disease that's in that Corynebacterium genus is the Corynebacterium diphtheriae, which causes the disease diphtheria. Now, one of the big issues of this particular bacteria is that when it grows at the back of the throat. It really causes issues with the respiratory system. It can cause this large growth called a pseudomembrane at the back of the throat. It's a toxin that this bacteria makes that kind of congeals everything to form this large growth. If this growth becomes large enough, it can actually cause death by suffocation because we can no longer get air. It grows back all the way into the airway that we can't even get air through the passageway. Diagnosing this particular bacteria, well, they can just visually look in your throat and see a pseudomembrane. That's pretty easy. If you don't have a prominent pseudomembrane, they can grow it on a specialized media in a lab called Loeffler's media. We can also do an antibody test as well. Treatment, they'll a lot of times give you an antitoxin. This is going to be that antibody that neutralizes the toxin so that pseudomembrane doesn't continue to form. They can give you antibiotics. Penicillin or erythromycin are easily ones that work. And then if that pseudomembrane is large enough that it becomes obstructive, they would surgically remove it. But, the best is just to not get these things in the first place, is keep up on your vaccines. That DTaP vaccine, the D, is targeting the immune system for the diphtheria toxin. So this particular vaccine is not making you immune to the bacteria, but it is making you immune and able to recognize the toxin that the bacteria makes, since that's the one that's causing the issues. So before we had this vaccine, we in the United States would have hundreds of thousands of cases a year of diphtheria, some of them deadly. Since the vaccine came out, and DTaP is one of the top, given vaccines, it's a pretty predominant vaccine, we've only had 12 cases of diphtheria in the past 15 years. Now, with declining vaccinations rate, we may see that go up. On our last and fourth group of our non-endospore gram-positive bacilli is the genus of Mycobacterium. And if you remember from lecture, we've talked about Mycobacterium because it's the bacteria that's in this group, and the Mycobacterium that have that mycolic acid in their cell wall. So they are gram-positive, but because that mycolic acid, which is a wax-like lipid, it does not gram stain accurately. So we have to do that acid fast stain on bacteria that's in this genus. Other unique characteristics, they are slow to grow. It just takes a lot of extra work to make that mycolic acid in the cell wall. If they do get eaten by our phagocytes, they don't get broken down by the phagocytes, so they can survive getting eaten. They can survive in those phagocytes so they can grow intracellular. They are resistant to lots of antimicrobial drugs. They're also resistant to heat, resistant to drying, chemicals, disinfectants. So just like when we are trying to disinfect against endospore-related bacteria, we're really also trying to target Mycobacterium because they are just hard to kill. They do require a higher strength disinfectant. Now, there are 75 species of Mycobacterium, but there's only 3 that affect humans. I mean, there's lots of organisms out there that affect different animals, plants, things that are in the soil. We only care about the ones that affect us, so we're only going to talk about the 3 Mycobacterium that affect humans. The first one is definitely the most common, and the most commonly and widely known is the Mycobacterium tuberculosis, which causes tuberculosis, a lot of times just shortened down to TB. Luckily, the cases of tuberculosis are decreased in the United States, but we do still have lots of cases that go undiagnosed and lots of people that may be spreading it without needing it or without realizing it. It is picked up through respiratory droplets and inhalation. What it causes once in the body. So if you breathed it in, someone coughed on you, and one of the symptoms is a cough, we breathe it in, and it can go through kind of 3 possible stages: primary, secondary, or disseminated. And I'm going to talk about them as I've got them on a picture on the next slide. So primary tuberculosis, this is when the bacteria, so here we have that initial Mycobacterium, it can live inside of white blood cells, and so as it gets eaten by our macrophages, it doesn't get broken down, and so you got lots of white blood cells that are like doing what they're supposed to do. They're eating up bacteria. But as they don't get broken down, all these infected white blood cells start to clump together until they form this large mass of infected white blood cells. And this big mass of infected white blood cells is called a tubercle. And so there it's called primary tuberculosis. This is when you just have these large tubercles in the lungs. But you have to remember what the lung tissue is there for. You have very thin cell walls so that you can have very efficient gas exchange. And if you have these large tubercles, and when I say large, I mean they're still small, but compared to how big some of these alveoli spaces are, they can really fill them up. It can really decrease that breathing, that it becomes hard, gasping, even sometimes for breath, because you just don't have that efficient gas exchange. So primary tuberculosis is when you just have all these tubercles, all these big masses inside the lungs. Secondary or reactivated tuberculosis is when you have these tubercles, these growths almost of our infected cells, and the cells start to die because our white blood cells aren't made to last forever. So now you start to have infected cells that are dying, and inside of them they're releasing the bacteria. And the bacteria may get released and head to another of our alveoli and then start the whole process over, or it may even get into the bloodstream. So this is when all of a sudden we're just rupturing the tubercle and it's starting to affect other areas of the lung. Disseminated tuberculosis is if it gets into the bloodstream and doesn't get targeted or destroyed by our immune system there, and it starts to spread to other parts of the body. So that last part, the disseminated tuberculosis, is when it starts to spread to other parts of the body. And the other parts of the body that seem to be the top causes of death from this bacteria is if it gets into the spleen, the kidneys, and even the brain. This is showing some images. These white masses in the lung tissues are these actual tubercles that are growing. So we can easily see this on x-rays and MRIs. Now, diagnosing it, well, they can easily take a sputum sample, do an acid-fast smear, and if you remember, they're looking for pink. I mean, although pink is kind of pinkish purple, as for acid-fast positive, that would tell you that it's a Mycobacterium. And of the Mycobacterium, those three, this would probably be the more common one that patients would have. They can culture and grow out the bacteria. They can do genetic probes and look for the DNA. They can also do skin tests and do chest x-rays looking for some of these massive tubercles in the lungs. You, depending on if you are working in a healthcare setting, you may have already had to have a TB test. I'm guessing you did not have this response with your TB test. What they actually do is they actually inject a very small amount of the bacterial antigen. It's not the actual bacteria. They do not give you tuberculosis, but it's an antigen. So a small, like, broken up section of bacteria. And if you've ever been exposed to tuberculosis, you have antibodies. And if your body's antibodies sees that particular antigen right there at that skin area where the injection site was, then you would have this big, huge reaction. That would say that at some point you've been exposed to it recently, that you have very active antibodies. If that turns out positive, then they do further tests, like do you have an active infection or was just a past infection. But it's a quick, easy glimpse at what's going on. Treatment, it's treatable. However, it's a hard bacteria to kill, so it's usually a combination of different antibiotics, some of the harder working antibiotics, and it's usually given for months, if up to 6 months even. You know, normal antibiotics, here, take this for 7 to 10 days. Well, this is take it, but for 6 months. Again, it's hard to kill. That extra waxy membrane makes it harder to kill. Prevention, we do have a vaccine called the BCG. It's named after the developers that made it. It's usually only given for those that are at a super high risk. Otherwise, face masks are one of the easiest ways to prevent passing that on and from picking it up. Our second Mycobacterium that can affect humans is Mycobacterium leprae, which causes leprosy. And it's not very common. I mean, honestly, I don't think I've really even heard of people with leprosy except in like the Bible. It's still around. Again, here in the United States, very rare. But if you're a traveling nurse and go somewhere, or you've had someone that's traveled, you never know. Maybe you'll see it. An interesting part is that this particular bacteria actually goes best in cooler areas of the body. So most bacteria that affect humans grow best at 37 degrees Celsius. Mycobacterium leprae grows best at 30 degrees Celsius. So if we're trying to grow it in a lab, we got to lower the temperature to grow it, which means it likes to grow and cause infections of the extremities. So your extremities, the tip of your nose, your ears, your fingertips are usually colder than other areas of the body. And so that's where it's going to grow best. Reason why I have armadillos up there is, guess what? Armadillos have a body temperature of 30 degrees Celsius. And so armadillos can actually be carriers of this bacteria. It is still relatively rare, though. I have heard of cases of it in the world still, but very, very rare. And not here in the United States that I've heard any time recently, and I would think it would make the news. How it is transmitted, person to person, usually a break in the skin, open wounds, and from nasal secretions. Again, rubbing your nose, not washing your hands. Treatable, just like TB, it's a combination of antibiotics given for a long period of time. And then our third and final of our gram-positive bacteria is the Mycobacterium avium-intracellulare, sometimes just called the Mycobacterium or M. avium complex. We're going to get into a lot of things that cause issues with AIDS patients because, again, they just do not have a working immune system. Of the Mycobacterium, of those 3 different species, This one is usually the most common Mycobacterium infection in AIDS patients. Healthy immune systems usually have no issues or any long-term effects from this bacteria. It's usually picked from ingesting contaminated food or water. This, just like the other Mycobacterium, can get inside of our cells and live just fine. And then once they do get inside of those cells, they'll eventually kill cells and cause damage anywhere they end up. This is showing a picture of some of these bacteria. I know they're showing pink. It's a different kind of stain. Well, it's acid-fast stain, but on my egg, it's very pink. But we can see all the bacterium that are actually inside of the cells. Ultimately, they will kill those cells. Just like before, treatment is difficult. It's combinations of antibiotics giving for long periods of time. It just becomes an issue though, because if you have no working immune system, the immune system can't even help with getting rid of the bacteria. It all has to be done using antimicrobials, really strong doses and for long periods of time. So there we finish up the last of the gram-positive bacteria. So you're ready to take the quiz, and I will see you in class next time.